morning. My name is Alfredo Castello, and I am principal investigator here in the Department of Biochemistry at the University of Oxford. So, I like to think about viruses as a fancy shell with a nucleic acid inside. So this nucleic acid basically will be delivered into the cell when the virus enters, and then will replicate, so multiply, and eventually will produce more viruses that will infect new cells. But some of the viruses that we see actually in TB infecting and being a threat for human health are actually made of a kind of nucleic acid called RNA. So normally we know a lot about DNA, but RNA is a very, very important molecule as well, and it has some peculiarities. So in the context of these viruses, they actually encode only for very, very few proteins able to interact and metabolize this RNA. And my hypothesis is that they should rely on cellular RNA binding proteins to basically complete this job, which is very, very complex, by the way. So the goal of my laboratory is to identify which cellular RNA binding proteins are required for viruses to actually spread, and to basically characterize how they work, how they do so. So I personally like very much HIV for these studies because it is a virus, it's a retrovirus, which means that it has an RNA genome that is reverse transcribed into DNA. And then this DNA will integrate into the cellular DNA. And from that stage, it will rely on cellular proteins, because even transcription to produce the RNA that will be packaged into the viruses will rely on cellular RNA binding proteins. So it's the paradigm of a virus that relies on cellular RNA binding proteins. So we use the CLARIOstar plate reader in multiple ways. One of them, and by the way, I forgot to say it before, so we do RNA binding assays with it, which means that we can actually, for example, express a protein fused to eGFP, and then cross-link it to RNA using ultraviolet light, which is a method used to study protein-RNA interactions. And then we can actually visualize if there is RNA or not by hybridizing with a probe that is labeled in red. So then by making a ratio between the red and the green, we can know if there is RNA binding or not, and in what extent. So another application that we have is because we establish all these cell lines expressing our candidate proteins fused to GFP, we can actually measure in real time how much GFP is produced and select the right times to do our infections. The third and main application, and I would say is my favorite one, is that we can actually use viruses that encode a red protein that basically glows in red, and we can infect cells and then monitor in real time how this red protein is produced, which is actually a proxy of how much viral protein is produced. So actually we can monitor in real time the infection using our atmospheric control unit, which is the key point to basically make the cells grow inside the CLARIOstar equipment. So I have to say it has many. The first one is the fact that it's very sensitive. So we have compared many devices, and we found it to be the most sensitive. Second, it has high flexibility because you can use different filters. It's actually monochromator, so we can measure at different wavelengths. This is quite interesting for if we are using different fluorophores, for example, or measuring multiple proteins at the same time, like green and red. Then it has, I think it's very solid equipment, so we haven't had any problems so far with it, so that's a very good point as well. And then the final and I think big point is the fact that the atmospheric control unit allows us to do these in vivo experiments with cells growing inside the equipment and measuring the fluorescence in real time. At this stage, we know which RNA binding proteins are involved in HIV and SIMBIS infection. What we want to do now is basically understand how they regulate infection. We have a multidisciplinary approach to do so, employing from virology to RNA biology, microscopy techniques, And in the future, what I want to do is to expand our analysis to many other viruses using a library of viruses encoding this red glowing protein to see which one has a role as a master regulator of infection. And this could be important because knowing that will allow us to develop drugs to target that protein and have a broad spectrum antiviral.